About 1 more minute. Okay, let's go ahead and get started. Let me get out of that, minimize that, and kind of drag it out of the way for you, Joe. So, thank you all. Welcome. It is my great pleasure to introduce you to Joe Girgente's master's thesis defense. And I'm AI. Okay, that's cool. I'm Nancy McIntyre. I am Joe's thesis advisor. And I'd like to first start off by recognizing the other members of Joe's committee. We have Dr. Chris Hill from Coastal Carolina University in South Carolina, and Dr. Stephanie Lockwood, who is with our TTU Waco campus. And thank you very much to all of the family, Joe's family, friends, guests, and other graduate students present here in the room who came out this morning to support Joe. So Joe, you may not know this, that Joe had a previous life as an engineer. He had a bachelor's degree in engineering from Virginia Tech, but realized that engineering was, but biology was what was exciting him. Biology was what was interesting to him. And so he went back to school and got a second bachelor's degree in biology, and then decided to pursue a master's degree to allow him to get a job actually enacting measures for conservation. So Joe started here in fall of 2019 with a background in surveying for aquatic invertebrates. And so he and I got on a conference call with two of the leading experts on dragonfly nymphs, which are the aquatic stage, Ken Tennyson and Marla Garrison, to try to brainstorm up a project for him to do within a master's time frame, something that was going to be novel, nobody else was working on, and doable within a master's time frame. And so that is what Joe is going to be talking about today with us with his thesis research.
All right, thank you, Nancy, for the introduction. For everyone out there, don't worry, I'll be limiting my talk today to just 1 or 2 hours, so just buckle up. We'll get through it together. As many of you know, I've been studying the ecomorphological variation in Hylogomphus geminatus in the Panhandle of Florida for the past 3 years, and I'll be talking today about what I found during that time. But I first wanted to begin by recognizing the following individuals and organizations listed here. The first of which is the Texas Tech University Association of Biologists, who granted me some money that I used to fund this research in part. The other organizations listed there helped me a lot with permitting for land access and also access to museum specimens. And the following individuals listed include, of course, my committee members, Drs. Nancy McIntyre, Stephanie Lockwood, and Chris Hill. My lab mates, past and present, Sean Suter, Justin Dawsey, Austin Biddy, Danielle Husband, and also Hannah, who happens to be my wonderful wife and has to put up with me every single day and helped me every day of field work, probably more than I helped her even. So very thankful to have her. And also the people listed under that last bullet point who are last but certainly not least, they helped from manual labor in the field to opening their homes to us while we traveled, to private land access, and also the expertise they were able to contribute to my project from the ground up, from its infancy. This research just really wouldn't have happened without these people present. So I'm very thankful for everyone listed here, and I'm sorry if I forgot anyone, including my honorary lab mates who are here today with us. So the twin-striped clubtail, Hylogomphus geminatus, is a small species of dragonfly that occurs primarily around the panhandle of Florida, and its original species description noted size discrepancies on either side of the Apalachicola River there, but no mechanism for that was ever proposed. So I wondered if it could be due to water quality. So this species, I'll refer to as geminatus going forward for the rest of today, is a member of the dragonfly and damselfly order, and these animals tend to spend most of their lives underwater as aquatic nymphs before they emerge as the winged adults we know today. During their time underwater, they are growing and developing, so they're exposed to variables that could impact their morphology. As adults, their growth and development is pretty much done, so any variables that are impacting them aren't necessarily impacting them in a morphological way. We do know that this species has a relatively restricted range noted by the gray shading here, primarily centered around the western panhandle of Florida. The blue line there represents the Apalachicola River, and they have possibly disjunct populations in Georgia and Mississippi. It's also probably semivoltine, which means that it spends 2 years underwater as an aquatic nymph before it emerges as an adult. And it can be considered a habitat specialist because it depends on these clear, shallow, sandy streams characteristic of the region. Now, these streams, you'll notice in some habitat shots as I go forward, are, like I said, clear, shallow, sandy. The sand is the main substrate here. It is the substrate on which the species depends. The nymphs burrow into it. And in other systems, they just really won't be there. So these clear, shallow, sandy conditions are the habitat of choice for this species, but they are also the habitat of choice for many other species in this region. This region is characteristic of hundreds or perhaps thousands of endemic flora and fauna that depend on these streams and nowhere else. So the pictures you see here are just a small fraction, a small percentage of the animals that are at risk in these regions. And if geminatus is potentially being impacted by some sort of water characteristics, then we can use it potentially as an early indicator of water quality issues that could impact not just it, but also all these other species that depend on this region and really exist nowhere else on Earth. However, we don't know much about geminatus right now. Its conservation status was last assessed in 2009. It was assessed as critically imperiled to vulnerable across the 4 states in which it's said to occur, but no data is currently being collected on that. So I developed some objectives to help answer these questions. The first of which is to, of course, determine the presence or significance of variations in the ecomorphology of both the nymphs and the adults. So, like the original species description said, is it truly larger east than west of the Apalachicola River? Of course, the next logical question, if those size discrepancies are significant, is what is causing them? Is it potentially some physical or chemical stream variables? Maybe some characteristics of land cover use, or perhaps something associated with the geographic location or the watershed in which they occur. And then finally, I also aimed to fill ecological knowledge gaps. We don't know, like I said, much about the species, and we don't really know how continuous its range is. There aren't a lot of behavioral kind of characteristics known about the species. However, for the purpose of this presentation, I'll be focusing on the first 2 objectives. If you are interested in some of the kind of anecdotal behavioral notes and other observations I made during the course of my research, I have those in my thesis, and I'll be happy to disseminate that information for those interested. So going forward, I'm going to break this presentation into adults and then nymphs. So we'll talk first about the adults, which includes the objectives and methods and the results for them before doing the same for the nymphs. And then we'll step back and look at it from a species level and kind of discuss as a whole what this means. So going forward, when you see this image in the top right-hand corner of your screen, you'll know that I'm speaking of the adults only. Again, recall objective 1 to determine the presence or significance of variations in its ecomorphology. And to do this, you need total lengths, of course. So I traveled to Florida and I measured 159 preserved museum specimens from the Florida State Collection of Arthropods in Gainesville, Florida, which you can see me doing on that image in the left. I also traveled to Florida twice per year and captured, measured, and released live adults, which you can see me doing on the right there. And in the middle is me rocking a mustache that my wife hated so much I had to include a picture of it. And that is a geminatus on my hat. It's not staged. It actually landed on me while I was in the streams, and I did stick my entire head into an insect net to capture that individual, and it is included in my data. However, to compare, I also measured 159 museum specimens of Hylogomphus obscurus, the common sand dragon, which I'll refer to as obscurus going forward. I did this because we wanted to know if any size discrepancies in geminatus were geminatus specific or perhaps characteristic of all dragonflies of that area. And I chose obscurus because it has very similar habitat to geminatus. However, it has a much broader distribution. So individuals measured from obscurus were selected from overlapping and adjacent watersheds as geminatus. So this species is one that is also characteristic of these sandy streams. I often find the nymphs and the adults alongside geminatus. And what I found when I ran Wilcoxon rank sum tests on the total lengths for both species on either side of the river was that both species showed significantly larger individuals east of the river than west of the river. However, geminatus was 8% larger in the east, while obscurus was just less than 3% larger on average. And these data and some other results are published in Girgente and McIntyre 2023 in the journal Insects. That is open access, so feel free to browse through that if you're interested. But we also noticed that there was sexual size dimorphism for geminatus, but not for obscurus. So the females were always significantly larger than the males with geminatus, but again, not for obscurus. And now that we've established significance, of course, the next question was what could potentially be determining that those significant differences. So I downloaded some data sets in order to use as predictor variables, and those included the 8-digit hydrologic unit code, or HUC8, watershed boundaries from the USGS National Hydrography Dataset, some land cover data from the Florida Natural Areas Inventory, and also some impervious surface data from the National Land Cover Database. And when using the HUC8 watershed boundaries as a predictor to examine pairwise watershed significance in the context of total length, I found that 18% of pairwise watershed comparisons for geminatus showed significant differences in lengths. Now, this is a complicated chart, so I'm going to break it down a little bit. On the x-axis are individual watersheds that are laid out on a longitudinal gradient much the same way they would be on a map. The vertical blue line represents the Apalachicola River, and it too is represented on a longitudinal gradient. So the watersheds on either side of it are actually the ones that occur on either side. And the horizontal dashed line is the average total length for all geminatus individuals measured. Significance here is denoted by unshared letters above each box plot. So if you cast your attention to the two watersheds on either side of the Apalachicola River, you'll note that they are represented by a D and an AB respectively. So this represents significant differences in size between those two watersheds on either side of the river. So not only were there 18% of pairwise watershed comparisons that showed significant differences in size, but there were also several adjacent pairs that showed significant differences in size, which is obviously quite interesting. However, when we did the same with obscurus, we found that there were only 4% of pairwise watershed comparisons showing significance, and none of those were adjacent to one another. There were also notable differences in land cover. If you see east of the river, there was proportionately more upland and sandhill habitat, which is really important for contributing the sandy substrate in the streams that geminatus depends on. There was also proportionately more wetland habitat, so potentially more suitable habitat for the species. West of the river, there was proportionately more rural and agricultural land. As you can see, over half of that land was represented by rural and agricultural influences. And impervious surface land cover really showed negligible differences, so we didn't include those data here. So to summarize, before we begin to talk about the nymphs, again, geminatus was significantly larger east than west, so too was obscurus, but geminatus averaged 8% larger, while obscurus averaged just under 3% larger. There were significant differences in more watershed comparisons with geminatus than there were with obscurus, and this included adjacent watersheds showing significant differences. And there were notable land cover differences east versus west of the river. So now I will discuss the same with the nymphs here, but we will basically do that before we, again, kind of back up and talk about what this means. So going forward, when you see this nymph picture in the top right-hand corner, you'll know I'm speaking about the nymphs. Again, recall objective 1 to determine the presence or significance of variations in ecomorphology for the nymphs now. To do this, of course, you need specimens. So I traveled to Florida and I used dip and kick nets in the streams for collection of nymphs. In addition to the ones I was able to collect, Dr. Ken Tennyson also loaned me a good number of his personal collection to use in my analyses. For that, I'm very thankful. So I ended up having 94 specimens that I could use for analyses that were associated with geographic location. Once the nymphs are collected, they must be preserved and measured. However, you can't just measure total length of a nymph because depending on how they're preserved, the muscles in the abdomen may contract or relax, which will then elongate or constrict the abdominal length. So total length is kind of relative to what the muscles do rather than any other individual factors. So we measure a different suite of standardized measurements that we do characteristically on these nymphs. And from the top in a clockwise direction, that includes head width, hind wing length, epiproc length, and prementum width and length. Once we have these measurements, we can classify the nymphs into instar, and instar is just the word we use to describe the age classes of macroinvertebrates. You do this by using a ratio of the hind wing length to the head width, but these data are usually species specific, so we had to plot them and figure out what those ratios would be for the different instars for this species. So the plot you see on your right shows 3 groups, age classes, instars, if you will, that were easily separated once the data were plotted. You can see in the top right-hand corner, there is that reddish pink F0 individuals. There were 23 of the largest nymphs, and we call these F0. These are the ones that will eventually emerge from the water and molt into adults, essentially. The next largest instar was the F1s, represented by 29 green dots there. And then we had 42 F2 pluses. Now, the F2 pluses are an age class comprised of several instars. This is where the ratio tends to overlap, and it's not easy to separate the age class. So you could have a large F3 overlapping with a small F2, for example. However, we generally want to run these analyses on F0s because, well, there are several reasons, but the 2 most important are F0s are the oldest. So they've been in the water for the longest. They've been exposed to whatever environmental factors are around them for the longest. So they would be the best thing to run analyses on. But also, studies have shown that they have a high relationship to the adult body size. So they are highly positively correlated with adult body size. A large F0 generally produces a large adult. And since we already know that the adults have significant size differences, it would be more interesting to do this with the F0s. But before we begin doing any analyses, we need to pick an actual response variable. Again, we don't use total length here. We have all these other measurements. So I ran correlation analyses to figure out if these variables were tightly correlated. And allometric relationships do hold true here for dragonfly nymphs, as we would expect. That means as an individual is larger, so too is its kind of body measurements in most cases. And I settled on using head width as a response variable because it's not only an easy measurement to obtain, but it's also tends to have not very much variation within instar. When I ran the same Wilcoxon rank sum tests on either side of the Apalachicola River for each individual instar group, I noticed that there was significance in the F0s and the F2 pluses, but if you are observant, you'll notice this is the opposite trend that we saw with the adults. So this is showing that Western individuals are significantly larger than Eastern individuals. And this is probably an unfortunate result of low sample size and sexual size dimorphism. So you can see there were only 5 and 6 F0 and F1 instars east of the river, respectively. And of the 5 F0 instars east of the river, 4 of them were males. Just like with the adults, the males are significantly smaller than the females. So you end up having an 80% sex bias size ratio in the F0s east of the river, which then results in some potential significance showing that they may be smaller in the east. For the F2 plus group, again, recall that this is comprised of several different instars, so significance may not really be indicative of very much here because it's possible we just collected more F3s east of the river and more F2s west of the river, for example. But because there was significance, I went ahead and ran some similar analyses to try to understand what might be causing it. So I again used the watershed boundaries, and I found that with the F0s, there was no pairwise watershed significance, and the same can be said for the F1s and the F2 pluses. So this kind of goes to show that perhaps the significant size differentiation on either side of the river may not really be indicative of anything on a watershed scale for the nymphs, at least. But I also recorded environmental variables at sampling locations where I was, and these included water velocity, turbidity, depth, width, dissolved oxygen, total dissolved solids, canopy coverage, water temperature, pH, and specific conductance. but I only collected four F0s at these locations. So the nymphs that were loaned to me from Dr. Tennyson were not associated with environmental variables because when they were collected, they were only associated with geographic location. So analyses using environmental variables had to be with just the nymphs that I collected. And because I had such a small sample size of F0s, I carried on without them and just used the F1s and the F2 plus. Again, I ran correlation analyses to understand which would be the best predictor variables. We don't want to use environmental variables if they're too correlated with one another, so we drop one of them typically. And what I found was that total dissolved solids and specific conductance had a one-to-one correlation, so I dropped total dissolved solids. I also eliminated velocity because it tended not to vary from site to site very much. And the scale at which it did vary was a little bit more fine than our equipment could probably measure accurately. And I also eliminated canopy coverage as a variable because I took that measurement at a single point in the stream, and it may not be indicative of perhaps the entire length of stream that the nymphs are using. With the remaining environmental variables, I used mixed effects models, using the environmental variables as the fixed effects and the site name as the random effect, and found that there were 3 significant environmental predictors for F1 head width. Those included dissolved oxygen, water temperature, and specific conductance. I'll talk a little bit more about them in depth soon, but note on this chart that these are the scaled effect sizes of the environmental variables on F1 head width. The blue symbols here indicate that there was a positive relationship, and the red indicates a negative relationship. When the same mixed effects models were run with the F2 plus group, there were no significant effects on head width. So to summarize, before we step back and discuss, again, there was some kind of questionable significance with the nymphs on either side of the river, but there was no watershed level significant differences. There were 3 significant predictors of head width for the F1s, and like I said, we'll talk about this more in a bit. And to kind of step back now and look at this on a broad scale. So what does this all mean for the species? Well, again, remember the east adults were larger than the west adults. There was also more suitable habitat in the east, perhaps proportionately, than there was in the west. So maybe there's some kind of habitat or landscape characteristics that are affecting the species, causing them to become smaller in the west than the east. And we also know that there were many more watershed level differences for geminatus, but not obscurus. So perhaps some sort of landscape effects are contributing to a watershed scale kind of effect that is impacting the geminatus populations, but not as much for obscurus. And the reason for this, I found out after doing these analyses, is that, like I said, geminatus is semivoltine, whereas obscurus is univoltine. This means obscurus is in those streams exposed to those potential chemicals for only a single year, while geminatus is in those streams for twice as long. So it is being impacted perhaps twice as much by anything in its environment. The mixed effects models, unfortunately, were statistically weak due to some low sample size issues. There was obviously not enough F0s to run them, but there also really weren't enough individuals east of the river for both the nymphs and the adults. So the results we found may not be indicative of a larger trend that's happening with the nymphs right now, but we did find those 3 significant predictors of head width, at least for the F1 instar. And I'll speak to these starting from the bottom up. So specific conductance is a known stressor in aquatic macroinvertebrates that causes reductions in assemblages and overall diversity. So it probably has effects on an individual basis. Water temperature is also an environmental predictor that has been shown in some studies to have a positive relationship with size, but there are other studies that note either a negative or a neutral trend as well. So this is another thing that is probably believable with our model. However, there, to my knowledge, is no literature that shows dissolved oxygen has a negative effect on head width or body size of aquatic macroinvertebrates. So this is something that could just be context specific here, and we might need to dig a little bit deeper on it to really understand if that is a significant predictor or not. And basically what this all means for the species is that essentially you have a restricted range already. And if you're seeing stressors that are causing smaller body sizes and potential extirpations across its range, that usually results in a lack of gene flow if those extirpations become broad enough. A lack of gene flow then would have a snowballing effect that could contribute to more and more loss of the species across its range. But additionally, these stressors can cause shifts in phenology. So individuals can fly at different times of year, which is essentially a fractured range anyway if they're not meeting. This species only has a 2 to 3 month flight window to mate and produce the next population. And if they're not meeting during that time due to differences in phenology, then you essentially have lack of gene flow again. And we can use this species, like I said, as an early warning sign to save other species potentially. So if we have a site like one on the left, and it turns into something like we see on the right, we know that something is wrong. We need to make sure that we're testing for whatever physical and chemical variables that could impact species of concern in that area. However, if something on the right is way upstream, we don't know. So maybe these impacts are washing downstream, and geminatus being impacted in a way that's causing it to become smaller is the early warning sign we need to say, hey, let's make sure we focus on these streams and help protect not just geminatus, but the other species within it. But how do we even know that that stream on the left is healthy to begin with? We really don't. So again, we could have point source pollution that is unseen. And if we're seeing geminatus that is significantly smaller in certain watersheds than in others, these are streams we really can focus on. So we don't have to spend time and money sampling all over. We can sample just individual streams or individual watersheds to understand if there are some kind of aquatic stressors in those streams. So to go forward with this similar research, I think there's a lot more we can do to answer these questions and to use geminatus as a more accurate early warning sign. First of all, we really just need a larger sample size of both the nymphs and the adults. That would solve a lot of problems, but that would also help us refine the mixed effects model. So I was testing for the presence or absence of environmental variables, basically, or using the presence or absence of environmental variables. But with the larger sample size, we might be able to test the interaction of these environmental variables as well. The adult analyses that I performed on geminatus and obscurus should probably be performed on other related gomphids, clubtail dragonflies, especially those that are semivoltine. However, we don't really know which species are semivoltine or univoltine half the time. So we really need more life history data on odonats in general. More quantitative habitat surveys could certainly give our statistical analyses a little bit more power. These could include prey availability, pebble counts, and community analyses. And increased environmental monitoring across a time span would really help. So when I measured environmental variables, it was a single point in time at a single place, and many of those variables were highly dependent on time of day or time of year. If we had data loggers in those streams that would allow us to use an annual average, then I think we could have more data that would be more useful in these similar analyses. And then the land cover analyses should be done on a watershed scale. So again, we can kind of see if there's a relationship between urban and agricultural lands and smaller geminatus on a watershed scale rather than just on an east-west basis of the river. And then we could also lab rear these individuals. Some lab rearing of odonats has been done. But when you do that, you can test specific environmental variables on the odonate development and morphology. So you hold the other variables constant. But really, all of this hinges on the untested assumption that size is relative to fitness in this species. There are studies on odonats that show size is correlated with fitness, but there, again, are studies that show that this is not the case. So this could be species or context specific. And we really need to know on the basis of size if this individual's fitness is being impacted. To do this, you could test some sort of fecundity analyses on females on the basis of size, or egg viability analyses, again, on the basis of size. But to go forward, that was what I was able to do in my time here, and I will take any questions. And I'll try and open the chat as well. So if you have questions, feel free to put them down in that chat. Yeah. All right. Any questions from the audience? Noria. I can repeat the question. Yeah.
How much is the water quality actually affecting its growth more than the species, because they belong to one species or another?
Yeah, so the question was, how does water quality potentially affect the growth of the species? So water quality obviously is a broad term. There are some factors that could affect the growth and development, while some may not. There are some species that are sensitive to certain things, but not to other things. There are studies that show certain things in the water, for example, water temperature impact growth by perhaps speeding up the growth actually, which then results in individuals that are smaller or larger depending on what they do after that growth period. With this species in particular, I do think that there are watershed level kind of water quality issues that are affecting the growth of the individuals and causing probably, I would assume that the F0s that result in smaller adults are also smaller as F0 nymphs as well. Unfortunately, like I said, I didn't have the F0 sample size to test that. But if I did, I think that's what we would see given some of the studies that show that F0 size is highly correlated with the adults. But yeah, as far as the actual growth rate of the nymphs, that can be wildly variable from what I understand and still result in nymphs of either equal body size to slower growth rate or smaller body size in some cases. There are different, I think each species has a different way of dealing with changes in growth rate. Thank you for all of the thanks. So, yeah. Sally asks if competition among different nymph taxa could be affecting size in the different systems. Sorry, that is also a good question and kind of why I wanted to run community analyses. I think would be really interesting. This is a small species of dragonfly in general, and it's a small gomphid dragonfly. There are other species in its streams that could certainly hunt and eat it. However, I think that the habitat it uses, so it's a burrower. It tends to burrow into the sand, and I think this is probably more to hide from larger taxa than it is perhaps to hunt for prey in that sand, because those regions tend to be not very high in diversity. So I don't think there's a lot of prey for it in those regions. But could the other larger individuals be impacting it? I would say perhaps. I kind of wonder if the adults are impacted more by larger individuals rather than the nymphs, because we've seen while the adults fly, there are also much larger species that tend to occupy that habitat, including one of North America's largest dragonflies, the dragon hunter, which is named because it eats other dragonflies, I think. And we've seen some instances where that species will come in and some smaller species will scatter. So certainly, I think it could be affecting that. Wade, thank you. Would there be any reason for prey quality, quantity differences across east-west gradient that may be driving size differences? Would there be any reasons for it? Probably. I think there was some differences. So sometimes we found streams where there just wasn't a diversity of macroinvertebrates in general. We might find geminatus in it, and we might not find much of anything else. And typically when we found that, it wasn't like we were finding a lot of geminatus. We were just finding one or two of them. But then every once in a while, we'd hit a stream that just seemed really productive. And there was just a lot of different taxa in those streams, potential prey, but also potential predators. The reasons for that, I'm not sure if that's perhaps something to do with dissolved oxygen or water temperature. But I think the presence and absence of the coarse and fine particulate organic matter sometimes predicted the diversity to an extent. Where streams were just sand, diversity seemed low. Where streams were just sand and fine particulate matter, diversity seemed low. But where we had a good amount of the coarse particulate organic matter, we tended to see more shredders and more scrapers and these things that are breaking that matter down. And I think these, as the kind of basis of the macroinvertebrate food chain, produced more predators and just a wider diversity of taxa in those types of streams. And I'd also add that the coarse particulate organic matter is probably, to an extent, a function of the kind of the amount the streams curve and the speed, right? If you have a channelized, very fast stream, it's washing away that matter. So you may not have much taxa there as well. Steve, water quality data and rapid bioassessment. It would be interesting to check if any of those data show the long-term water quality trends you suggest may be important. Indeed, I think I did look at in the early stages to see if there were like long-term water quality data on the scale that I would need it. I think that if there are those data present, My assumption is that they would not be across the spatial scale necessary for the analyses, but certainly would be interesting on a stream-to-stream basis. And that's probably something that can be done going forward for sure. Yeah, Chris mentions it's a lot of work kicking through those streams. Kicking through streams is part of, one of our favorite things to do. So we'll do it every day. Have you considered, instead of landscape scale, local effects? It look like you have quite a different substrate east versus west. It's going to influence prey, but also predation. Ignoring the sex bias in sampling, is it possible that there are differential drivers east versus west? Reaching refuge size quickly is valuable for nymphs in west, but perhaps competition is more significant driver in the east. Yeah, those are great questions too. And I kind of thought about that to an extent. So something that came up when I was writing my thesis was the level of competition between large and small individuals in that species. So if you have large individuals, hypothetically, they should be able to displace smaller individuals in male-male competition at least. So wouldn't it make sense that large males are moving around, displacing smaller ones, and spreading those large genes if it's on a genotypic basis? So if they're displacing those smaller genes, then we should see either mostly large or similar sized individuals, but we do in fact see these differences in size, which to me kind of said that even if genes are flowing, then the size of the adults is potentially not a genetic factor, but a factor of their growth and development in the streams based on some kind of environmental inputs. And could there be different drivers east versus west? Absolutely. I think there's probably different drivers on even a smaller scale than that, on a watershed scale, of course. What those different drivers are, we can really only speculate right now. I could come up with 100 of them, but I don't know that I could label any one to any specific region, for example. But again, if perhaps the urban and agricultural influences are driving body size in the west, but not in the east, then it's like Chris said, it could be maybe competition that's driving it in the east instead, something like that. Yeah, organic matter. Why is there such a high amount of endemism in the region? That's a great question. I've wondered that too. I think the endemism in the Panhandle region is probably due in part to the geologic processes that created that region in the first place and caused it to have some different substrates and different kind of patterns in the landscape. That is kind of the southern extent of the Appalachian Mountains, and in some of those streams you can tell. There's also a lot of really cool steep heads, which Hannah will talk about in a week, and other spring systems and cool characteristics there. And I think that contributed to potentially just some novel landscape and novel microhabitat that allowed species to really expand and speciate. And that would be my assumption, but I'm sure there's people that know way more than I do about that. Any other questions from the audience or the Zoom?
I have one that was emailed to me.
We have a question via email. I'll repeat.
Is it true that people?
I think my parents emailed a question in, and the answer to that is no, as far as I understand. As far as I was told from Bill Nye the Science Guy. Another from Steve. Do you know of any other species that have a size difference between east and west of the Apalachicola River? It seems like there could be a historic reason for genetic differences in populations. Another great question. We did look into that at some point. I looked for species with size differences, not just on either side of the river, but some kind of weird, distinct, like adjacent size differences. And I really couldn't find much. Most of those things seem to be a function of someone's already looked at it and turned that into a subspecies or something like that. Perhaps, maybe geminatus is subspeciating. And that was kind of an interesting question I had too, but we would need obviously more than just the size is different to be able to analyze that. So I do not have an answer to that, unfortunately, but I do wonder if there are perhaps some amphibians or reptiles, probably amphibians, that would exhibit some of those size differences, but I couldn't find anything. How far do the adults disperse? Great question. Again, something that's not really known and why we need more life history data. However, This is a lotic species, so it occurs on moving waters. And typically with the lotic species, we found that they have a smaller dispersal distance than the lentic species. Shout out Steve and his dissertation work. But the lentic species typically disperse farther because their habitats are ponds and whatnot, which are subject to drying up. So they need to have the ability to move to other locations where they can lay eggs again. For these lotic species, they may not disperse out of their watersheds. We don't know. But the fact that there were significant size differences in adjacent watersheds to me was still interesting because I would assume that some of the individuals are dispersing out of their watersheds to other streams. But because we don't know this, we don't know that anything really about their dispersal. We didn't use the adults in any association with the actual like environmental variables at the streams. Something we thought about was, well, could we use water temperature to predict the adult size? Well, again, when you capture an adult at a stream, you don't know if it came from that stream or if it came from far away. So we didn't really have environmental variables to use with the adults. So sorry, that didn't really answer your question, Doug.
Other questions from in-person or Zoom? Okay, well, join me in thanking Joe for a very interesting presentation.
Thank you. Thanks to everyone who tuned in. You missed out. We had coffee and donuts here. And to my committee members, Stephanie and Chris, we will reconvene here soon. We're going to head up into Nancy's office, and we will be using the other Zoom link, I'm told. But check in the next 10 to 15 minutes if you have any issues.